Hi, this is Mr. McLean, and in this session, we're going to talk a little bit about active transport. We're going to focus in on the sodium-potassium pump and why that's such a critical biological function. Okay, I'm going to turn my camera off at this point, and I'll come back later. All right, so what we saw in the last video, we were talking a lot about what it means to be passive, and passive just means that the direction of movement is based on a concentration gradient. So if you were using proteins to make that easier, to facilitate it, it would still be passive as long as it's going down the gradient. And we've looked at a couple different examples of that. Now, there are some instances where we work in the opposite direction. So instead of going down the gradient, we actually want to work against the gradient. And that's where active transport comes in. So it's moving against the concentration gradient. So in other words, we're going to be building up a greater concentration of a particular substance or ion on one side of the membrane compared to the other. So in order to do that, you're working against the natural movement of those molecules. So if you want to kind of pump them into an area, you're going to rely on investment of energy. And in this case right here, we're using ATP. So that's going to be our main energy molecule for most of the processes that we examine in this course. So the first thing I really want to focus in on is the sodium-potassium pump. And as I mentioned in class, the sodium-potassium pump is of immense importance, and we use about a third of our body's ATP to drive the sodium-potassium pump. So when we start with this particular image right here, what we're going to see is that embedded in our phospholipid bilayer, we have this transport protein, and that is the sodium-potassium pump itself. And the way that it's configured, whenever it's open to the cytoplasmic side, what we're going to see is that there is an affinity for sodium ions. Okay, so for this little cartoonish sketch, what we see is the sodium ions kind of have that circular configuration. So it is open and ready for sodium. And one thing you absolutely want to know is that this particular protein is designed to take in 3 sodium ions. Okay, so it's designed to work and move 3 sodium ions across the membrane. And we're going to take that from the cytoplasmic side out to the extracellular environment. So in order for that to happen, we need to change the shape of this. And what we're going to notice is that the investment of energy, which comes in here as the form of ATP, and it's going to drive the change here by phosphorylation. So that's right there by adding the phosphate to it. And basically what we've done is we've taken our adenosine triphosphate, so it's got those 3 phosphates, and we sever that 3rd phosphate off. So when we remove that phosphate right here and attach it on there, that physically will cause a change that does this. Okay, so we went from here to here, and now the affinity for sodium is not good. So the sodium is going to be released, and we're going to see that those 3 sodiums are now moving outside. At the same time, the configuration is now favorable for potassium, and you're going to see that there's only 2 spots for potassium. So in this next image, we have 2 potassium ions that are going to bind. And once the second one binds into this position, what we notice is the release of this phosphate takes place. So we release this phosphate off of here, and as a result of that, our protein does this. So it goes from here to here, and now it's in a configuration where potassium will be released, and at this point, we're back to the beginning. So when you look at the sodium-potassium pump, you might at first wonder why this is so important. And we're going to show you that as we kind of progress through this, and I'll give you a quick example of an area. But I also want you to notice that there's actually two things that are being generated as a result of this sodium-potassium pump. And I'll throw a slide in here just so we can kind of talk about this. So if this is our membrane, and not real easy to draw on here, sorry guys, but here's my membrane. And then on the inside here, let's just say our protein's kind of configured like this. Okay, so as we use that sodium-potassium pump to mobilize those two ions, what we're going to end up doing is we're going to build up a massive concentration of sodium outside here. Now, the sodium doesn't want to stay outside, and we're doing the same thing over here. We're building up potassium ions on this particular side. Okay, and the potassium doesn't want to stay here. So each of them really want to follow their own gradient. They want to kind of squeeze back through there, but they're ions, so it's not easy. They have to utilize a channel. Okay, and this channel is gated. So we're going to talk about how voltage gated channels will keep the sodium pretty much locked in up there. And the same thing's true about the potassium. So there's potassium channels that are also gated. Sorry for the sketches, they're not perfect. But once again, in this configuration, neither the sodium nor the potassium is able to restore back to its own gradient. So we're actively building this up. The other thing I want you to notice, if this is the outside and this is the inside, in fact, we can go back to one of the pictures. Maybe let's use this one here. It's a little bit easier. If we look at this, what we've done is we've moved 3 sodiums for every 2 potassium. So we have a 3-to-2 ratio working there. And what that's telling me is that I'm going to end up with a voltage. And the reason why I have a voltage is I have a difference in charge across the membrane. So if we look at it, the interior is going to become negative in regards to the exterior because 3 of those positives have been moved this way where only 2 positives have been moved that way. So it's created a differential which has made the exterior positive in relation to the interior. So we got 2 things happening here. Number 1, we have a chemical concentration gradient. And number 2, we have a voltage. So when you put those 2 things together, what we end up with is an electrochemical gradient. Now, this slide right here just explained what that voltage is. And we call that a membrane potential in this case right here. And a membrane potential is a voltage difference across the membrane. So that voltage was created because we have a difference in how we distribute those ions. So by us moving 3 out and only moving 2 in, that helps create that difference. So whenever you put those 2 forces, the chemical concentration and the electrical difference, you get what's referred to as the electrochemical gradient. Okay, and that is going to be really critical as we move on and we study things like the nervous system. So from the standpoint of an animal, probably the most important process following this particular type of active transport is the sodium-potassium pump. Okay, and from the standpoint of plants, they utilize what's called a proton pump, which is literally moving hydrogen ions. So when we look at the thylakoid and we see the accumulation of hydrogen ions that are taking place, that's going to be an example of where we see a proton pump. Now, I'm not going to run it in this particular section of the video, but I've shown you how the neuron is reliant upon the sodium-potassium pump. So when we think about it, our nerve cells, let me see if I can get myself a blank slide in here somewhere. There we go. All right, so our nerve cells, I'll just draw it like this. This is my lipid bilayer. Okay, these are lipids. And so here's the outside, here's the inside. And what we find is that we start to get a net negative charge on the inside, and we have a net positive charge. So if we look along the length of our cell here at different stages, the inside's negative, the outside's positive. When we get a disturbance in our plasma membrane, so we get a stimulus, sort of like you flick your finger or you bump into something, what that does is it starts to open up some of those sodium channels. So if we were looking at what's happening with our voltage, if we were to graph it, our voltage of our cells, if this is 0, our voltage are negative, and then at a certain point, if we get enough of a disturbance, we get an influx of positive. So what we're going to see is these sodiums are going to go rushing in here. And you don't necessarily need to know the details of this. And I would definitely recommend looking at a video of the action potential. But the idea is, once we see that flooding of positive sodiums, we get a depolarization. So you can see right here on my graph, I was originally a negative value. Now I've shot up to a positive value for the inside of the neuron. And then in response to that, our potassium channels open. And as the potassium starts to flood, what we notice is we see a charge once again becoming negative inside there. And then there's a little bit of a recovery. And the recovery in getting us back to what we call resting potential. In other words, the charge when our neuron is at rest, that is accomplished by the sodium-potassium pump. So if you think about it, if you had some type of a stimulus that occurred and you were unable to restore the original charge, then you would not be able to you would not be able to see that type of transmission of a nerve impulse again. Okay, and then here's an example of a proton pump. So in this case right here, we're using energy and we're building up a massive gradient of protons, which are hydrogen ions. Okay, the last thing I have in this section here, hopefully the battery doesn't run out, is we have co-transport. And if you look at this situation right here, what we're seeing is we're using ATP to build up a hydrogen gradient using a proton pump. So I got all these hydrogens. The hydrogen naturally wants to diffuse. As the hydrogen diffuses through this co-transport protein, there is not only a spot for hydrogen, but there's also a spot for sucrose. And what we're going to do is we're going to be able to move the sucrose only when we move the hydrogen. So by building up a lot of hydrogen, we can therefore transport not only the hydrogen back through, it's almost like you have to have two keys in order for the thing to open up. So by us building up this hydrogen gradient, it allows us to mobilize that sucrose. Okay, and that's what a co-transport is. So the idea is active transport of a solute indirectly drives transport of another solute, so that we can actually see substances moving into the cell. And hopefully that was helpful. We'll examine a few other points of active transport in another video. Thank you.